The third and final speaker in session 3 is Professor John Blenis. John is a professor of cell biology at Harvard Medical School and certainly one of the world's experts in the area of S6 kinase and mTOR signaling. John.
Yeah, 2. Okay. I'm ready. Okay, so I want to thank Cell Signaling Technology and MIT for sponsoring this event, and especially Mike for really organizing this. Really great, a great day of science. My lab has been working for many years on these pathways here. Let's see, I want to do this. Oh, this is a test. Here we go. On the Rna Helicase pathway, Pi3 Kinase mTOR pathway. What I'm going to do today is really focus on one study in our lab, which is focused on the role of mTOR, S6 kinase, in regulating various aspects of cell metabolism, cell growth, and survival in cancer cells. Now, before I start, those of you who were here this morning and saw Steve Gigi's talk, you know, if you're like Wade and I, we see Steve quite often. We never know when he's being very serious or not. So I decided to go investigate. I went down into the men's restroom, and sure enough, there's Bob, and he's thinking about some algorithms and looking at protein-protein interaction studies. Now, this was a long time ago, and so they made a movie out of this, and they actually brought in this new actor and bring this. So, all right, so let's go to Tor. Okay. So I'm going to focus on the Tor pathway, and this is what I really consider as a central part of the Tor pathway. This is the tumor suppressor TSC1 and TSC2. It acts as a gap for the GTPase Reb. It shuts down Reb. When you turn off this gap, then Reb gets activated, directly activates TORC1, and then TORC1 plus a variety of other signals activate S6 kinase to signal downstream to regulate pathways that are important for cell growth control and cell proliferation. Now, the pathways become extremely complex over the last 15, 20 years, and it's impossible to tell you all the complexities of the pathway. But I kind of break it down into two general areas. One I loosely call the ancient part of the pathway, and the other I call the more recently evolved part of the pathway. So this is the more ancient part of the pathway, and I can say that because several components of these inputs are conserved back into yeast and flies. These are the amino acid sensing pathways that's been worked on predominantly by David Sabatini here. And then there are the glucose sensitive pathways. We still don't know actually the mechanism of how this works. The glucose sensitive pathways where AMP kinase is a critical component of the pathway, but we recently published a study where an AMP kinase 1 and 2 minus cells, this reveals another mechanism for energy sensing in the cells, and that's through energy-dependent regulation of this complex, TTT-rubel, which helps to assemble a dimeric activated TORC1. And then there's the oxygen sensing pathways that many labs here over at Harvard and Penn and other places have been working on over the years. Now, layered upon this more ancient part of the pathway are the more recently evolved tyrosine kinase pathways, and they include the Pi3 kinase pathway that many labs have worked on to show how they intersect with this box to regulate TORC1 signaling, including work from Brendan Manning and Luke Cantley's lab and Andy T. in my lab many years. They published several papers looking at this connection. And then Philip Rue in my lab also has looked hard at how RISC can regulate TORC1, and he's done that while he was in my lab and also in his own lab. He's found many connections. Now, one of the things I want to point out here is that in this pathway, you'll notice that these guys in green are known to be overexpressed or activated in some way, and that these all contribute to TORC1 signaling. And these guys in red here, and these guys are putative tumor suppressors. So these tumor suppressors, when you have loss of function of the tumor suppressors, you get hyperactivation of the TOR pathway. And it's believed that this pathway is activated in about 70 to 80% of all cancers to varying degrees. Okay, now in addition to the upstream signaling we have been working on for many years, what's happening downstream of mTORC1, and I didn't define this, but assume most of you know mTOR exists in two complexes. mTOR, when it's associated with Raptor, forms the TORC1 complex, and mTOR, when it's associated with Richter and SYN1 forms the TORC2 complex. I'm going to focus on TORC1. TORC1 is activated. It regulates a variety of downstream signaling processes summarized here that are involved in various aspects of metabolism and regulation of autophagy. And I'm going to really focus on these areas of downstream signaling. So TORC1 regulates all these biological processes, regulated by all these upstream inputs, activated in a large number of cancers. And so having a drug for mTORC1 you would think would be a very therapeutically beneficial drug. And in fact, there's this natural product isolated from bacteria, soil bacterium in Easter Island that is a very potent antifungal and a very potent inhibitor of TORC1, does not affect TORC2. So that was the picture 10 years ago or so. Now we know that rapamycin does not efficiently inhibit all TORC1 signaling. Very efficient in inhibiting S6 kinase, but other targets are not as dramatically affected. And in fact, rapamycin in clinical trials has not been as successful as we had hoped in the beginning. It has some effects, oh, this is wonderful. It has some effects on some tumors like renal cell carcinoma, but on others, there's no effect. And part of the reason is it's cytostatic, and part of the reason why it's only cytostatic, even when it's most effective, is that there exists these feedback loops and crosstalk pathways that are engaged when you inhibit TORC1 that generates part of the resistance mechanisms. There's many more mechanisms, and many of us are looking at that right now. All right, now, I'd like to turn that light off, but I don't think I'll be able to. I'm going to focus first on protein synthesis. This is a movie that many of you have seen, and I like to show it because it really summarizes some work that we've done on the contribution of mTOR and S6 kinase to protein synthesis. We've published several papers on how TOR regulates various aspects of translation control, from pioneer rounded translation, which is a splicing-dependent mechanism, to translation initiation at the cap, as well as fine-tuning translation through the regulation of S6 kinase and in this complex here, this process here. So let me just start off by saying that Marina Kaufman-Holtz, a graduate student in the lab many years ago, did some affinity purification with S6 kinase, a TAP construct that she generated. And she got lots of really cool things, and none of which she wanted to follow up on. She found also many subunits of the EIF3 complex, especially the 5 core subunits, would always come down with S6 kinase. And I just thought that was garbage. But she was smart enough to buy some antibodies without me knowing, and did a series of immunoprecipitation experiments that are summarized in this. So she did immunoprecipitation experiments, glucose gradient centrifugation experiments, cap binding experiments, and so on. One other thing I want to point out is that in addition to the core subunit and the other associated proteins, there's another EIF3 complex protein called EIF3J. And that's, that association John Hershey has shown is regulated by TOR signaling, although the mechanism is not known. But that's important because Tatiana Pestova has shown that 3J is critical for bridging the gap between this scaffolding protein and the 43S complex, the ribosomal complex, 40S ribosomal complex. Okay, so what you want to imagine here is that you have starved cells and you're stimulating with nutrients and growth factors. And what happens by these co-IP experiments was that, what Marina found was that when you stimulate cells with nutrients, all of a sudden mTORC1 will bind to the EI3 scaffold complex. Now it's in proximity to phosphorylate S6 kinase at a single site, 3 389. That's not sufficient for activation, but it causes dissociation. Because if you mutate that to alanine, it doesn't dissociate. If you mutate aspartic acid, it's always way. 3J binds. Oh, this is not active, but Dario Leslie showed that PDK1 will bind and phosphorylate and activate the enzyme. This then forms a complex that binds to the cap of the mRNA. Now TORC1 is in proximity to phosphorylate 4-ABP1. This is a translational repressor. It dissociates from the cap. This now creates a docking site for another scaffolding protein called EIF4G. So 4G will bind to the scaffold, this complex, and it does a few things. It helps to stabilize the complex, stabilize this large complex. It also brings in additional proteins, and I can't even see it. It's under here somewhere. But there's this poly-A binding protein that binds to the 3' end of the message that helps to circularize and stabilize this translational apparatus. And then this protein there, EIF4A, right, is a really critical enzyme. This is an RNA helicase. And this is important for unwinding the structured mRNAs and a small number of mRNAs that play an important role in cellular bioenergetics, cell cycle progression, and angiogenesis. However, this helicase is not active without its regulatory subunit that should be here, EIF4B, and it's also suppressed by this tumor suppressor, which I know you can't see. Oh, wow. PDCD4. All right, now I can't mess around. Okay, so we have this complex, and this is actually translationally effective, but it's not very efficient because this helicase is not activated. All right, so now S6 kinase is activated after TORC1 phosphorylation and phosphorylation by PDK1, and there's some other events. There are 9 phosphorylation sites that are important for regulation. S6 kinase is not done, and it phosphorylates ribosomal protein S6, the original target of S6 kinase. We still don't know really what that does, but it correlates with translation initiation. But it also phosphorylates this regulatory subunit EIF4B. This is that subunit that's important for 4A activity. Phosphorylation to that site recruits it to the complex, and Marina showed that if you mutate it to alanine, it doesn't go. You mutate it to aspartic acid, it's there predominantly. But the helicase is still not active because there's this tumor suppressor which inhibits the helicase called PDCD4. But S6 kinase is not done. It phosphorylates PDCD4. That creates a docking site for beta-TRCP. You get ubiquitination. This is Michele Pagano's work, and this is degraded. And now you have a translation initiation complex, pre-initiation complex, that has an active helicase that improves its ability to efficiently translate messages that have these highly structured 5' UTRs. Again, things like HIF1 alpha, c-Myc, PDGF, VEGF, FGF, GLUT1, all proteins involved in bioenergetics and cell cycle progression, cyclin D. Okay, so when you have activation of these proteins or loss of function of these proteins, you're really driving TORC1 signaling. So TORC1 signaling is driving protein synthesis at many levels. That was just one little small aspect of how it regulates translation. And the important thing to know about this is that when it's driving all these biosynthetic processes, the cell is eating up ATP. And any important signaling system, you've already heard this throughout as a general theme, that these signaling systems that play critical roles often do many things to coordinate everything, to keep everything normal, or as normal as possible. Okay, so when TORC1 is driving this, consuming energy, it also then turns on pathways that we assume it was turning on pathways that will allow for increased nutrient uptake and metabolism to generate the energies to support these processes. So the questions that we asked were, what are the nutrient sources for energy and carbon building blocks? And then how does TORC1 regulate the metabolism of these nutrients? Okay. So a few years ago, Andy Chu, graduate student in the lab, published this paper which showed that in cells that have high TORC1, they're dependent on both glucose and glutamine for survival. If you inhibit TORC1 in this context, you can take away glucose and glutamine and the cell will survive. They don't grow, they just sit there, but they survive for a while. So that says that when you have high TORC1, you absolutely need glutamine and glucose. And in fact, if you reduce glucose in these cells and you inhibit glutamine metabolism, again, these cells will die. Not in the presence of rapamycin, though. Okay, so in addition to rapamycin therapies, we want to also identify therapies that take advantage of the fact that these tumors have high TORC1, so a synthetic lethal type of approach. Okay, so the question we next asked was, does TORC1 regulate the metabolism of glutamine to produce these carbon building blocks in ATP? And if it does, what is the mechanism? That's what we're all about here. What is the mechanism? And does this reveal an Achilles heel for mTORC1 driven tumors? So this is the TCA cycle and glycolysis and fatty acid metabolism. And PPP is in here somewhere. And what do we know about TORC1 and metabolism? I want to say that a lot of the experiments I'm going to tell you about over the next few slides are completed in a colon carcinoma cell line, or in TSC2 null MEFs, or in TSC2 null LAM cells, and prostate cancer cells. But we'll focus on these here. Okay, so what we know about TOR is that it does contribute to glucose metabolism. We know that MYC and HIF-1 alpha are involved in regulating many of these steps in glucose metabolism or glycolysis, and there's been a link between TOR and MYC and TOR and HIF-1 alpha, although the mechanism is not really clear, and I'll address that later on. We also know from work from David's lab and Brendan Manning's lab that TOR can regulate fatty acid biosynthesis and sterol synthesis. And so the question that we wanted to ask is TOR regulating this step? Does TOR regulate the metabolism of glutamine, glutamate, alpha-ketoglutarate to replenish the TCA cycle to drive the production of these building blocks and also to drive production of ATP through oxidative phosphorylation? Okay. So we first looked at the effect of glutamine uptake, of rapamycin on glutamine uptake in these cells that have high TORC1. When you add rapamycin, glutamine uptake is decreased. Then we asked, what about intracellular glutamine levels and also glutamate levels? And this just shows you the glutamine result. When you add rapamycin, the intracellular glutamine levels accumulate. So we're reducing the uptake of glutamine, but we're accumulating intracellular glutamine. And that suggested to us that we must be inhibiting the metabolism of glutamine. Even though there's less uptake, there is accumulation of glutamine here and glutamate to a lesser extent. Okay, so we then went on to collaborate with Sarah Fent and Gregory Stephanopoulos' lab, and then we did some isotope tracing experiments with Sarah just to look and determine whether in fact glutamine was being a source of TCA cycle intermediates. And I'm not going to show you the data, but it's summarized here. So when we add rapamycin in these cells, we see a decrease in all of these products in the TCA cycle downstream of glutamine. And so therefore, TORC1 does regulate glutamine anapyrosis, the refeeding of the TCA cycle through glutamine. Okay. Now, there are two major enzymes that we were interested in. That's glutaminase and glutamate dehydrogenase. We looked at the several other enzymes that could potentially be involved here, like the transaminases. We didn't see much regulation, so we really focused on these two enzymes. So the first thing we did is to look at the protein levels under various conditions when you have high TORC1 or low TORC1. So in these cells, these are TSC2 null cells where we put TSC2 back into them. And we've also done this in other normal cells. We see very low GLS levels, glutaminase levels. However, when TOR is on, the GLS levels go up, and that increase in protein production is sensitive to rapamycin. We saw no effect under these conditions on GDH protein levels. Okay, so there's decreased GLS protein, and then we also, because S6 kinase is downstream of TORC1, we wanted to know if S6 kinase was involved, and this just shows you results from an S6 kinase inhibitor. We've knocked down S6 kinase, also an overexpressed RAP-resistant S6 kinase, and we see the same result. So that says that TOR, through S6 kinase, is regulating GLS expression. Okay, so how does this occur? What's the mechanism? How can we find the intermediates in this pathway? Well, I showed you that S6 kinase is involved, and what we already knew, and I mentioned earlier, is that MYC may be downstream of this pathway. And what was already known was that MYC is upstream of GLS. And the way this works is Qi Dang showed that MYC suppresses transcriptionally the expression of the microRNAs 23A and B, which regulate the expression of GLS. So there were lots of data suggesting a connection between these two and this guy, and so we wanted to know what the connection was. And what we already knew, based on some work in Brendan Manning's lab and based on work with Junying Wang's lab that we collaborated with her, was that S6 kinase can do two things. It can regulate, it can suppress it can induce MAD phosphorylation, which induces its degradation, and it can also inactivate GXK3's ability to turn over c-MYC. But our data suggested there must be something else in addition to these connections. And so we went on to study this in a little more detail and took advantage of the fact that what I just showed you in that movie was that S6 kinase, we know S6 kinase is involved, plays an important role in regulation and translation of messages of highly structured 5' UTRs like c-MYC. And we had all these reagents, these 4A, 4B reagents, PDCD40 reagents to test that hypothesis. Okay, so we went on about to test the hypothesis. If 4B, 4A is involved in regulation of c-MYC. And this is important because there are other RNA helicases that can be involved in this process. Okay. So this just shows you that, again, when we knock down S6 kinase 1 and 2, like when we inhibit TOR signaling, we see a decrease in glutamine consumption. So that's import, and then the intracellular glutamine levels go up with the compound that inhibits SS kinase, also with knockdown of SS kinase 1 and 2. Okay. This just shows you that a MYC reporter looking at transcription downstream, and we show that if you add mTOR inhibitors or SS kinase inhibitors, you block MYC-dependent transcription. And this is an important experiment where we put in ectopically expressed c-MYC, and we show that rapamycin no longer affects GLS levels, shown here. Then finally, we wanted to ask, okay, what about, are these guys really involved here? And so what we did is simple experiments that everyone does nowadays, is that we knock down EIF4B, or we knock down EIF4A, and when we knock down either 4B or 4A, we see c-MYC go down and GLS go down. And this is an important experiment for this model, is that we mutated, remember I told you if you mutated that phosphorylation site to aspartic acid, that it's always in the translation pre-initiation complex. So we express that protein, we add rapamycin, and we rescue the effects of rapamycin on both GLS and c-MYC. And then finally, Tom Roberts' lab, Haosheng Tong and Tom Roberts' lab generated a 5' UTR connected up to a reporter construct, and we showed with that construct that, again, rapamycin and the SS kinase inhibitor would block the translation efficiency of this reporter. Okay. So, in fact, what we think we've done is we've bridged this gap between mTOR1, SS kinase, EI4B4A, and c-MYC. And this is an effect on translation efficiency, so you don't get rid of it completely, but the cell is really smart, and it uses other mechanisms. Any protein that's left are inhibited by loss of, are suppressed by MAD1 phosphorylation, dephosphorylation, or by GSK3 inactivation in this pathway. So multiple ways of regulating c-MYC, which all converge onto the regulation. Sorry, I'm pointing to which regulate the expression of c-MYC, or activity c-MYC and expression of GLS. Okay, now again, this whole idea of metabolism, if a cell wants something done, it targets it at multiple steps. So now we asked, okay, we've figured out a way in which TORC1 can regulate glutaminase levels. What about glutamate dehydrogenase levels? Does TOR regulate GDH? Okay, so the first experiment we did was actually look at flux between glutamate and alpha-ketoglutarate. Again, this is with Sarah Fent. I'm just going to remind you that we have no effects on GDH levels, and I showed you earlier. But we did see an effect on flux between glutamate and alpha-ketoglutarate when we inhibited TORC1 with rapamycin. Then, because it wasn't affecting levels, we looked at the activity of GDH, and we find, in fact, and this was a collaboration with Seung-Min Jeong in Marcia Hagase's lab. They helped us with the assay, and we find that GDH activity, in fact, goes down when you inhibit TORC1 signaling. So, now we have the question of how does TORC1 regulate GDH activity? Okay. We know that TORC1 doesn't directly phosphorylate GDH. And this took advantage of the fact that Marcia Hagase had previously shown when she was in Linda Garanti's lab that the SIRT2 and SIRT4 or SIRT4 can directly inhibit GDH activity by ADP ribosylating activity. This is independent of its deacetylase activity. Okay. And this just shows you that in cells that have high TORC1, SIRT4 is repressed. If you inhibit TORC1, it goes back up. So if you inhibit TORC1, this goes up and now can inhibit GDH. And this is just done in duplicate here. And this just shows you in the DLD1 cells, we see the same thing with rapamycin. SIRT4 goes up. And in this case, SS kinase is not involved. Okay, so in the case of glutaminase, it's mTORC1 and SS kinase. In this case, TORC1 is doing something independent of SS kinase to regulate glutamine metabolism. Okay, so how does TORC1 regulate SIRT4 levels, this inhibitor of GDH activity? I'm not going to go through this. This just shows you that it's a transcriptional effect. When you have high TORC1, you have low SIRT4 mRNA, and when you inhibit TORC1, SIRT4 mRNA goes up. And therefore, GDH activity is regulated through TORC1-mediated control of SIRT4 transcription, unlike the TORC1-mediated control of GLS, which is translational. So we have TORC1 upstream of SIRT4 through a transcriptional mechanism. But we still were not done. We still needed to bridge this gap. So how is TORC1 regulating SIRT4 transcription? There are about 20 or so potential transcription factors that could regulate SIRT4 expression. We went through those. Fred Seeby, I forgot to mention this is all Alfredo's work. He found that one of those looked like a good candidate. So he looked at its phosphorylation status in cells that have low TORC1 or high TORC1. and he saw a mobility shift. This is sensitive to phosphatases, and then it was also inhibited by rapamycin. This is with insulin and serum stimulation, and this is with amino acid stimulation. He wanted to know what phosphorylation does. And what phosphorylation does is it destabilizes the protein. If you inhibit TORC1, the protein becomes stabilized. And he showed that this destabilization was dependent on beta TRCP, dependent ubiquitination and degradation. So we have TORC1 phosphorylating CREB2. This results in its degradation, can no longer induce SIRT4. SIRT4 goes away, then you activate GDH and you promote this step in glutamine metabolism. Okay, I'm not going to go through these slides because I think we're, because of time, but we have lots of data that suggests that SIRT4 has tumor suppressor-like activity. I'm just going to run through these real quick. If you overexpress SIRT4 cells, these DLD cells, they grow a little more slowly, but importantly, if you put alpha-ketoglutarate into the mediate, you completely rescue it. So you rescue the effects of overexpressing SIRT4. Tumor growth is reduced, tumor volume is reduced, and the proliferation of the tumor cells is greatly reduced when you overexpress SIRT4. Importantly, also, and then George Pouligianis from Lucantley's lab did some bioinformatic analysis for us, and he finds that many cancers, SIRT4 levels are in fact down, and this is just a matched tumor, normal and matched gastric tumor, and we see also SIRT4 levels are down. And importantly, Rna Helicase's lab has provided a lot of evidence to suggest, in fact, with mouse models that SIRT4, in fact, is a tumor suppressor. Okay, now, okay, I'm going fast, I know. Looks like I have about 5 minutes. All right, so I told you at the beginning that, you know, we have TOR and we should be targeting TOR, and certainly we should continue targeting TOR. We have problems with rapamycin in that we induce many feedback loops and mechanisms for resistance, but it turns out that rapamycin and rapamycin analogs in combination with other therapeutics is being very effective in mouse studies, and maybe even we'll hear some more about some clinical trials in combination with other agents. So certainly rapamycin is still going to be a very useful drug, most likely in combination therapy approaches. But what about targeting cancer cells that have high TORC1? Leave TOR on, so it's driving these metabolic processes. Can we target those metabolic processes so that they will induce death in cells that continue to promote these anabolic processes and consume energy? And so that was the idea that we've taken in trying to understand other ways of killing TORC1-driven cancers. And I'm going to tell you one little story, then we have some others that we're working on, other combinations. Okay, so this is just a proof of concept, all in vitro tissue culture cells, and it's still yet to go into a mouse model. In this case, remember earlier that I said that when you reduce glucose levels in cells that have high TORC1 and you inhibit glutamine metabolism, those cells die. They won't die if you add rapamycin because you need to be driving anabolic processes. So let's leave TOR on. But we can't really take away glucose from patients. So how else can we interfere with glycolysis? And there are many ways you can do that. We just took a really crude mechanism where we add DNA damaging agents, which we've already heard will affect metabolism. And it's been shown that some DNA damaging agents like nitrogen mustard will in fact reduce glycolysis. And we confirmed that by doing metabolic analysis, and we see that production of pyruvate is reduced in another target. I can't remember what the other metabolite we looked at. Or reduced when you add low doses of this DNA damaging agent, doses that don't really kill the cell, okay, in these cells. Okay, so with that in mind, we've wondered if we block glutamine metabolism, will that have an effect on cells that have high TORC1? So we're reducing glycolysis, and now we're going to attack the glutamine metabolism pathway. And what this shows you is one experiment with this compound, natural product from green tea. It's called EGCG and inhibits GDH. We've also done this with a compound called BPTES, which inhibits the glutaminase step. We've also knocked down these enzymes, and we've also just taken away glutamine from these cells. And what we always see is that in the presence of low-dose nitrogen mustard, inhibiting glutamine metabolism synergizes to induce death in these cells. And importantly, this is specific for cells that have high TORC1, because if we put TSC2 back into these and reduce TORC1 signaling, you see no effect. And even more importantly is that this is completely due to reducing input into the TCA cycle, because we can rescue this effect by simply putting pyruvate alpha-ketoglutarate, OAA into these cells, and they survive this combination. And we don't just see it in cells with TSA2. We've looked at other cells, the colon carcinoma cells, prostate cancer cells, and a few others. This just shows you another model system where we looked at P10 null MEFs. The combination has an effect. If you put P10 back in those cells, it has no effect. Okay. So what I've shown you today is that when you have lots of nutrients, lots of growth factors and nutrients, or you have activating mutations of upstream components or inactivating mutations of tumor suppressors, you can have high TORC1 activity, and this drives anabolic processes to make carbon building blocks to support cell growth, survival, and proliferation in these cells. In addition, what I've shown you is that TORC1 can also drive metabolism like glucose, and we focused on glutamine metabolism by Alfredo's work, and you're driving these processes to generate the energy and the carbon sources needed to support the ability of TORC1 to drive these processes. Under conditions where you have lots of nutrients and growth factors and you have high TORC1, if you have too much energy, this is like having too much money. You can only buy so much. And then what do? You reinvest that money to make more money. And this is what these tumor cells do. They reinvest the energy to further stimulate TORC1. And as I mentioned, it can be done by regulating AMP kinase, which is a negative regulated pathway, so high energy will suppress this. And we've also just recently published this, what I mentioned earlier, is that when you have high energy, you stimulate the formation of this TTT-rubal complex, which is required for stimulating the generation of a functional dimeric TORC1 complex, which further increases metabolic pathway. So this is positive feedback loop. Okay, so I'm just going to end. So we've identified new pathways linking mTORC1 to glutamine metabolism, which reveal therapeutic vulnerabilities. These are the people involved: Alfredo Sebi, Jeannie Lee, Sang-Gyun Kim, Andy Chu, who's no longer in the lab. He's actually now working in New York City at McKinsey. And then we have some other people in the lab that are now contributing to these, to our understanding of mTORC1 sleeping. So thank you for your attention, and I guess we can talk at the break.